This lesson is on regulation of transcription. We're going to look at just the very basics of how eukaryotes regulate whether a gene is read out into messenger RNA, whether it is transcribed. So this dark line here is meant to represent a single strand of DNA. This is going to be our template strand. This is the 3' end over here. This is the 5' end over here. And again, this is the template strand that we're going to be using as a template to make a copy of when we make messenger RNA. It's also called the antisense strand. And the other strand, the one that's going to have a nucleotide sequence that's identical to the messenger RNA, except the messenger RNA is going to have U's instead of T's, we call that the sense strand. That's going to be up here, and it's not shown. Now remember that the region where the RNA polymerase binds, the sequence of DNA where the RNA polymerase binds, is called the promoter. So that's a non-coding sequence of DNA where the RNA polymerase is going to bind, and it has a directionality, and it's only going to be on one of these strands. It's only going to be on the antisense strand. So it determines the direction that the RNA polymerase is going to go. So for example, here, the RNA polymerase that I'm showing here in red is going to go in this direction because of the orientation of the promoter and the way that it binds. And it's also telling the RNA polymerase which strand is the template strand. So RNA polymerase is going to move in a 5' to 3' direction. Now don't get confused by looking at the template strand down here. What we mean when we say it moves in a 5' to 3' direction is that as it lays down new RNA nucleotides, it's adding them on the 3' end. And so the 3' end is the end that's growing, and so it's moving 5' to 3' relative to the strand that it's laying down. So we've seen now that there's this one non-coding sequence of DNA, the promoter, which is where the RNA polymerase binds, but there are other regulatory DNA sequences that are important in regulating transcription as well. For example, enhancers. Now, an enhancer is a region of DNA, and it can actually be quite far away from the promoter. I'm not showing it that far away here, but it's a sequence of DNA where different kinds of proteins, here I'm showing one in green, called transcription factors can bind. Now, a transcription factor is a protein, and you can have different kinds of transcription factors. For example, you could have activators, which bind to enhancers, and as the name activator and enhancer suggest, they're going to make it more likely that this gene is going to be transcribed. So they are activating or enhancing the transcription of this gene. You can also have repressors. Repressors do the opposite. They can bind to regions of DNA and prevent the transcription or decrease the likelihood of transcription of that gene. Or they can also just interfere with activators binding to enhancers. So we've got these regulatory DNA sequences like enhancers, and then we've also got floating around these transcription factors. They are proteins like activators and repressors, which can bind to the regulatory DNA sequences and make it more or less likely for the gene to be transcribed. So it's the interaction of these two, the transcription factors binding the regulatory DNA sequences, that helps to determine whether or not we're going to get transcription. Now, how does that happen? It's not at all obvious from the way that I've drawn it. But what's actually thought to happen is that the DNA loops around. So it's not like shown here, but when the transcription factors bind, the DNA loops around, and so the enhancer, for example, ends up here, and the transcription factor over here. Let me draw that in green so it matches what I did before. And then it's going to bind other proteins that actually help the RNA polymerase to bind to the promoter, and therefore make it more likely that transcription is going to progress in this direction. So the transcription factors bind to the regulatory DNA sequences. They recruit other proteins that help the RNA polymerase bind to the promoter, which then starts polymerizing RNA. It's the interaction of all of these things which can get complex, which is going to make it more or less likely for the gene to be transcribed. Now, just to give you some examples of things that can be transcription factors, hormones can bind to receptors inside cells. For example, that's what steroid hormones do. They bind to receptors inside cells, and then the hormone receptor complex itself becomes a transcription factor binding like we've seen above. We can also have intracellular signaling molecules that can do the same kind of thing. So there's a whole variety of different kinds of proteins that can bind to the DNA to make it more or less likely that transcription will happen. So that's a very brief introduction to regulation of transcription in eukaryotes.